My name is Lan from Agriculture and Agri-Food Canada. I will be presenting on the analysis of sample types and sequencing methods for optimal characterization of the honeybee gut microbiome. Honeybees pollinate a variety of plants, including crops of economic importance, such as apples, blueberries, and canola. Honeybee productivity is dependent upon their health, which can be influenced by a bacterial community that resides in the gut, known as the microbiome. The honeybee gut microbiome is a conserved community that consists of up to 9 core members. Most of the bacteria are found in the gut, which is the region circled here. The gut microbiome can be altered by biotic and/or abiotic stressors, and therefore can be used as an indicator for bee health. The goal of our work is to compare how sampling and DNA sequencing methods affect the detection of the honeybee gut microbiome. To determine the best approach for studying the honeybee gut microbiome, 4 sample types were isolated and sequenced: the whole bee, the abdomen, the whole gut, which again is the circled region, and the hindgut, which is the end segment of the gut. For each sequencing experiment, 10 individual bees, as well as 1 pooled sample, which contained a fraction of each individual sample, were analyzed. To compare how sequencing methods affect the identification of the microbiome, each sample was analyzed using 2 sequencing methods. One approach was 16S rRNA amplicon sequencing, as seen in this figure on the upper right. This method specifically targets a region in the 16S ribosomal RNA gene. The other approach was metagenomic sequencing, which is seen here on the bottom right. This method sequences all that is present within a sample. We compared sequencing at the shallow level, which corresponds to a coverage at less than 40 million reads, and at the deep level, which corresponds to coverage at greater than 40 million reads. The raw sequencing data was processed using the program Kraken-2, which allowed for taxonomic classification, as seen from the figure on the left. Using the same program, we were also able to conduct rarefaction analysis, which allowed us to have a look at species abundance, outlined here on the right. 5 gut microbiome members were consistently detected for each sample type, as seen on the x-axis, regardless of the sequencing method, which is shown on the y-axis. The bacteria included Lactobacillus, Gilliamella, Freshella, Snodgrassella, and Bifidobacterium. The box plots show the distribution of the 10 individual samples, as well as the pooled sample, which is the red circle. The pooled sample generally falls within the distribution, which suggests that it is representative of the individual bees. Next, we looked at how different databases affect the detection of the gut microbiome. Here, shallow metagenomic sequencing data was analyzed using 3 databases, the top panel showing B-Rolama, our in-house database, the middle using the NCBI nucleotide database, and the bottom showing the Silva database output, which is used for 16S rRNA analysis. At the genus level, the B-Rolama and NT databases provide similar results. However, the detection levels are very low for all 5 genera using the Silva database. Therefore, database selection for metagenomic sequencing analysis is crucial for optimal output. Rarefaction analysis was run on our classification results to visualize species discovery rates for each sample type. On the left is the analysis for the individual samples. When shallow, seen on the left facet, and deep, on the right facet, sequencing are compared for the different sample types, as shown on the far right. Taxa are discovered more quickly with deep sequencing than when compared to shallow sequencing. This is seen for both the individual and the pooled samples. Therefore, metagenomic sequencing improves taxonomic resolution. The increase in information gained from deep sequencing allows for in-depth gene discovery and functional profiling. For example, as seen here, we identified several bacterial genes involved in carbohydrate metabolism. We showed that pooled samples can represent the core microbiota of individual bees at the genus level. We also showed that database selection is crucial for the optimization of data output. Untargeted metagenomic sequencing provides greater taxonomic resolution compared to targeted sequencing, which allows for gene identification and functional analysis. With that, I'd like to thank the many contributors to this project, including our funding sources. If you have any questions, please feel free to contact me. Thank you for your attention.